Hello everybody, welcome to this third part of the video-based tutorial. Here we will briefly look into a simple cancer simulation so that you can get a better sense of how one can really simulate cellular systems that do something and at least understand the basics of the coding approach. We're going to have a look at a very simple simulation of a cancer where each cell essentially just grows and divides recursively. So one cell at the beginning grows and divides, and then the daughter cells again grow and divide, and so on. And let us have just a brief look at the code of this model. So when we look here in the source folder of the CancerSim1 project, then we have these two files, one .cc and one .h file, and in the .h file we have implemented the behavior of these cancer cells. So if we look at CancerSim1.h, then you can see here in the Sublime editor or whatever editor you like, we have here again the simulation function similar to before, where we instantiate the simulation in the first line, and then we initiate the resource manager, and then we create a cell object. So the cell class is the most basic cell class that we have, and it's in the standard library of BioDynaMo. We assign it a certain diameter, in this case 30 microns, and we then add a so-called biology module to this cell. So a biology module specified what some element in the simulation does, some biological element. It doesn't need to be a cell element or a cell body. It can be also, for example, a part of an axon or a part of a dendrite, or of a blood vessel. This is really up to the modeler, and there are different biology modules, so depending on what they do, they can have different names, and some of those are already implemented in the standard library, like for instance grow divide. So grow divide is a biology module which, as the name says, grows and divides, and there are two main parameters here. The first parameter is the threshold for division, so at which diameter does the cell body divide, in this case 32 microns. And the second number here is the speed at which the cells grow, the cell body grows, or the volume grows of this element. And in this case we use this number of 3000, and then we add this cell to the resource manager, and then as before we simulate it, in this case with 3000 time steps, and if that works then we should see this message in the terminal. So let's have a look at this simple example running. So I go here to the CancerSim1 project folder, which is the one that includes this source file, and then I simply type BioDynaMo build. That was very quick. And then BioDynaMo run. And that takes now longer, also longer than before, because now we have many more cells. At the beginning we have one cell, but then it increases. Don't be distracted by these messages here, which is because of this remote desktop application. I think it causes some kind of issue, but it doesn't really affect the behavior of the simulation. And now importantly, well, first you can see that the simulation was done successfully, and then also you can see here this bdm.toml file. So if we have this file, if you look at it, you can see that it specifies the visualization file. So it's actually a file that is required to specify what is what is exported and what kind of features of these cells or characteristics of these cells are exported and at what time interval. And this is also mentioned in the online documentation on the website of BioDynaMo. And in this case, we export all the cells and we export also the diameters of those cells. Now, if this has run correctly, then we should be able now to visualize this simulation. So I will start ParaView, which is what we are using to visualize our simulations. And here you now see that this window was opened. And by the way, this should be installed also once you have done the normal BioDynaMo installation. Then as a dependency, this will also be in the standard installation script also be installed, ParaView. And now we can to go back here. So we go to load state, and then we go to output, KansasSim1, and now there should be a file called KansasSim1.pvsm, and this is the file that we need to load, where all the information that we listed in the TOML file should be included. And we just increase a little bit the size now of the window here so that we can better see. Then we can also zoom out a bit. So we have zoomed out. So now you see the initial cell here. And if we here click on play, then you see how this cell divides and grows, and the same happens with all the daughter cells. And you can also scroll, turn around and look a bit or explore this simulation while it is happening. And so you can see that quite a large number of cells gets created in this way. And just to a little bit better understand the workings of ParaView, we here have this pipeline browser, and if we look at the cells item here, then you can see different aspects or features of this cells visualization, and one of them is this diameter characteristic that we had exported. And so that's why we here see really the diameters changing, because we take this into account. So I would suggest that you have a bit of a look at this simple and basic simulation and the visualization, and then play a bit around with this. You can maybe try out different growth rates or different kinds of thresholds, and then you can try to get a better understanding of how to run and execute such a simulation and then visualize it, because of course visualization is a very important part of such scientific simulations. Alright, then see you next time.